In this proposal, we will be exploring upregulation of SPRED2 and repression of TRKB, ERK, MAPK pathway activity as a therapeutic approach for OCD. Obsessive-compulsive disorder is a neuropsychiatric disorder characterized by a recurrence of intrusive thoughts, referred to as obsessions, followed by repetitive compulsive behaviors or compulsions. OCD typically follows a cycle of an obsession, which leads to anxiety, and is then followed by a compulsion that leads to temporary relief. Some of the issues in treatment for OCD are a lack of diversity in pharmacological therapies, given that as of now most drugs are SSRIs, which means serotonin reuptake inhibitors, or augmentation drugs which increase the efficacy of these SSRIs. These drugs typically target the same serotonin and dopamine systems, demonstrating the limits of drug therapies available. Another issue in treatment is drug resistance in patients with OCD. One way in which these two issues in treatment merge is in an example where BDNF polymorphisms have caused drug resistance to fluvoxamine, which is an OCD drug that is used as treatment, and it's an antidepressant. This ultimately sheds light on the importance of searching for new drug therapies that can target both of these issues and result in better treatments for OCD and a wider variety overall. Given that BDNF polymorphisms have been associated with OCD drug resistance, it seems like a possible target for drug therapies and research. Some of the pros in that are that it's been identified as a potential effector in many neurological disorders, including OCD. However, some findings have been contradicting across various studies. While some studies have linked increased BDNF mRNA expression levels in patients with OCD, other studies have found no association between levels of BDNF and OCD symptom recovery after treatment with fluvoxamine or risperidone, which are two OCD drugs. Therefore, a possible compromise might be to instead target one of the main pathways BDNF interacts with, which is the TRKB, ERK, MAPK pathway, which I will be referring to from now on as a TRKB pathway for simplicity. BDNF is a substrate to this pathway, and it binds to the TRKB receptor, which results in a conformational change or a set of conformational changes that then activates the MAPK and ERK pathway, as we can see highlighted here on the right. The protein SPRED2 would be a perfect option given that it negatively regulates the TRKB pathway and it activates downstream MAP kinases. And most importantly, it's also a link between the pathway and OCD, as the same study that linked OCD behaviors with TRKB pathway activity actually did so through a SPRED2 deficiency. So in this study, they essentially knocked down SPRED2, as we can see here, and this ultimately led to OCD behaviors in mice, where the mice engaged in excessive grooming over long periods of time as well. And it also led to an increase in BDNF TRKB pathway activity, which can be seen here on the right, as the knockouts have much higher rates of phosphorylation of TRKB and phosphorylation of PERK, or ERK, but PERK is the phosphorylated version. Thus, these findings ultimately emphasize how SPRED2 is a gene that's a novel therapeutic target for OCD, as it plays a role in the pathogenesis of OCD and has a regulatory relationship with the pathway. Therefore, as we propose making SPRED2 the target of our research, our first hypothesis is that SPRED2 interacts with TRKB, ERK, or another known kinase in or near the pathway. And our second hypothesis is that the overexpression of SPRED2 causes a decrease in TRKB pathway activity in OCD and decreases OCD symptoms, which is based on the rationale that when the gene was knocked down, it increased pathway activity and led to OCD behaviors. In order to conduct our studies, we will be using the SAPAP3 knockout mouse model, which is an OCD model. In this study, they knocked out the gene in mice, and this overall led to OCD behaviors, On the right here, this graph and this graph, both, they essentially show increased lever press rates that were identified as being habit-oriented in the knockout mice instead of goal-oriented as they were in the wild-type mice, which ultimately was established and categorized as OCD behaviors. So in order to tackle the first part of our hypothesis, our first aim is to characterize the role of spread2 in the TRKB pathway activity in OCD. We will do this through immunoprecipitation assays, and we will conduct Western blotting in order to identify potential binding partners of spread2 within the TRKB pathway. We will also be doing immunoprecipitation assays in conjunction with mass spectrometry in order to identify potential binding partners of spread2 near the pathway but not included in the Western blotting. And overall, our goal would be to explore the mechanism by which spread2 represses the pathway activity and how this plays a role in OCD. So in order to tackle our first aim, our experimental design would look something like this. We would use wild-type and OCD mice, and we would yield amygdala cell lysates from tissues in their brains. And we would then conduct immunoprecipitation assays in which we use bead coupled with anti-spread2 antibody in order to pull down anything that is bound to spread2. And when we do that, we would be pulling down the spread2 binding partners. On the left here, we would be doing Western blotting using antibodies for ROS, TRKB, BDNF, and ERK, as those are some of the most important proteins in the pathway. And aside from that, we will also be using these extracts in mass spectrometry in order to identify any other known kinases that might be playing a role in phosphorylating spread2 or potentially binding to it. Our potential results might vary. However, if Western blotting reveals that some of the binding partners are TRKB and ERK, or if mass spectrometry reveals other known kinases as binding partners, some of our future directions would be first to question, does the kinase phosphorylate spread2? And then to also question, is the kinase involved in the repression of the pathway by spread2? And if so, how? So other further studies should be done in order to figure these things out and use these as kind of stepping stones to learn more about spread2 and its mechanism in the pathway. In tackling the second part of our hypothesis, our second aim is to measure the effects of spread2 overexpression on the TRKB pathway and OCD behaviors. As a reminder, we want to overexpress spread2 because a previous knockdown of the gene showed an increase in TRKB pathway activity and also OCD behaviors. We will conduct the overexpression through CRISPR and DNA sequencing. And then in terms of measuring the effects of this overexpression, we will be conducting phosphorylation assays and Western blotting in order to measure changes in the TRKB pathway activity. And we will also be conducting reward devaluation assessments in order to measure the changes in OCD behavior. Our ultimate goal would be to determine whether spread2 can be used as a target to develop a new drug therapeutic approach for OCD. So the first part of tackling AIM2 would be to overexpress spread2, and we will be using CRISPR to do so. Our first step would be to inject CRISPR reagents into zygotes, and our DNA would basically contain the spread2 gene and ECR1, which is an enhancer that will specifically overexpress spread2 in the amygdala of the mice, as that has been shown in previous studies to be a central place where OCD is highly affected or impacted. The next step will be to confirm the spread2 overexpression, which we will do through DNA sequencing once the mice have grown into embryos. And when that is confirmed, we will then implant the embryos into mice surrogates, which will yield our wild-type mouse and our treated OCD mouse. The colors don't represent the color of the mice, but they're just more for clarity. So in order to test the effects that the overexpression has on the TRKB pathway activity, we'll be using wild-type, treated OCD, and untreated OCD as a control mice in order to yield amygdala cell lysates from their brain tissue, as we've done in previous parts. We will then use these cell lysates to conduct phosphorylation assays and Western blots. In our first assay, we will be targeting ERK expression, so we will be using antibodies that'll be anti-ERK1 and 2 and anti-PERK1 and 2. This one represents the phosphorylated version of ERK, which would show that the pathway is active. And then we will also be doing the same thing with TRKB, in which we will be using anti-phosphorylated TRKB Y1515 anti-TRKB to note the normal protein expression unphosphorylated, and then anti-GAPDH as a control. In addition, we will be doing RT-qPCR and BDNF as a follow-up in order to determine whether spread2 affects BDNF levels as a way of repressing the pathway. This is what our expected results would look like if our hypothesis is supported based on previous studies that conducted pathway activation tests. So in the wild-type and treated OCD, we would see that there is a very low amount of phosphorylated ERK1 and 2, but very high amounts of unphosphorylated, which would reflect not much activity in the pathway. And here in the untreated OCD, we would see high levels of phosphorylated ERK, which would ultimately highlight that the pathway is very much active. Similarly, down here, we would see in the wild-type and treated OCD, not very much amounts of p-TRKB-Y1515, which is the phosphorylated version, but larger amounts of the TRKB unphosphorylated, which would show that the pathway is not active, whereas in the untreated OCD, there would be high amounts of phosphorylated TRKB since the pathway will be highly active. And overall, these results would be supporting our theory that spread2 overexpression would lead to a decrease in TRKB pathway activity. So in order to then measure the effects that overexpression of spread2 had on OCD behaviors, we will be using the wild-type, treated OCD, and untreated OCD mice as a control. And first we will train them through an RI paradigm in which they learn to press a lever for a sucrose reward. We will then conduct a devalue test where they're fed sucrose before, and then they're also allowed to press the lever whenever they want a reward. And then a value test, which overall controls for satiety, which is the level of fullness in the mice. So they'll be fed chow before and then allowed to press a lever for a sucrose reward. The goal of this test is overall to determine whether the actions of the mice are habitual or goal-oriented. And it's expected that the wild-type mice would engage in goal-oriented behavior, whereas the OCD mice would engage in habitual behavior. So if our results support our hypothesis, we would observe very similar behaviors in the wild-type and treated OCD mice, in which they would have higher lever press rates per minute in the valued part because their activities would be goal-oriented, whereas in the devalued parts, they would be lower since they're being fed chow beforehand. And here in the untreated OCD mice, we would see very similar levels, which would ultimately reflect habitual actions instead of goal-oriented, which is associated with OCD. And here on the right in the devaluation index that was created in the study for the SAPAP3 mice, we'd ultimately see that the wild-type and treated OCD have a devaluation index that is closer to 1, which indicates that it's wild-type behavior, whereas untreated OCD mice would have a devaluation index closer to 0, which indicates that it is OCD behavior. Thus, in terms of future directions, after our studies, we would want to confirm the observed patterns in other established OCD mouse models, not only in the SAPAP3 knockout mice that we will be using. We would also want to use identified spread2 binding partners that we found in our first aim as targets of future studies, and to further explore the mechanism by which spread2 represses the TRKB pathway, since that could prove to be an ultimate way that we could actually transform this into a drug therapy for OCD. And here we have an overall summary of our aims in this proposed study. Our first aim, again, is to characterize the role of spread2 in TRKB pathway activity in OCD, which we will do by identifying potential binding partners of spread2 within any of the TRKB pathway, with the ultimate goal to explore the mechanism by which spread2 represses the TRKB pathway. And then our second aim is to measure the effects of spread2 overexpression on the pathway, as well as OCD behaviors, which we will do first by measuring pathway activity, and then also by measuring changes in OCD behaviors, with the ultimate goal of determining whether spread2 can be used as a target to develop a new drug therapeutic approach for OCD. And once again, here we have our 3 different mice that we would use in our experiment. And thus we conclude here with our references and are excited about the outsets of performing these proposed studies in order to find new drug therapeutic approaches for OCD.